the most unique thing about planet Earth, shared by no other known planet. Life on Earth is incredibly vibrant and diverse. This is because of DNA, sometimes called the blueprint of life itself. But how does
Golden Corrals carved New York strip and jumbo butterfly shrimp make me feel like a real New Yorker.
You see what I'm talking? Antonio Meucci invented the telephone and he got robbed. Everybody knows that.
But how does DNA create everything we see around us? Well, it's through a process called protein synthesis.
Protein synthesis is a critical process when we look at the actual function of DNA in organisms. It is divided into two parts. First, there's transcription, and next,
Golden Corrals carved New York strip and jumbo butterfly shrimp make me feel like a real New Yorker.
You see what I'm talking? Antonio Meucci invented the telephone and he got robbed. Everybody knows that.
So first there is transcription and then translation. These two processes are then further divided into three phases each, beginning with initiation, then elongation, and finally termination.
First, DNA goes through transcription, the process of converting DNA into messenger RNA, or mRNA.
Transcription starts with a phase called initiation. During initiation, an RNA polymerase will bind to a section of DNA known as the Tata box, which is a specific sequence of DNA nucleotides. The Tata box will tell the RNA polymerase where in
Golden Corrals carved New York strip and jumbo butterfly shrimp make me feel like a real New Yorker.
You see what I'm talking? Antonio Meucci invented the telephone and he got robbed. Everybody knows that.
and in which direction to move down the DNA.
Next, transcription will go through a second phase, known as elongation.
During elongation, the RNA polymerase will unzip the double helix of the DNA. It will then move down the strand and build a complementary RNA molecule. Except for an RNA, the T or thymine base is replaced with a U or uracil base. So adenine will pair with
Golden Corrals carved New York strip and jumbo butterfly shrimp make me feel like a real New Yorker.
You see what I'm talking? Antonio Meucci invented the telephone and he got robbed. Everybody knows that.
I'm not going to keep doing this. I'm not even getting paid. Okay? Uracil. Adenine will pair with uracil, and cytosine will pair with guanine.
Eventually, the RNA polymerase will code a termination sequence. Then the RNA polymerase will code a few more nucleotides and eventually sever the mRNA molecule from the DNA.
After these steps, the mRNA is not yet ready for translation. It first needs to be streamlined.
The mRNA contains sections of it that are irrelevant. These are called introns and don't actually contribute to the phenotype of the organism. Other regions, called exons, do contribute to the phenotype. The mRNA will bind to proteins called spliceosomes, and they will cut out the introns and fuse the exons together.
After removal of these introns, the mRNA can undergo translation. Initiation starts when the mRNA binds with a ribosome.
Ribosomes are composed of a large subunit and a small subunit. These are made up of rRNA, or ribosomal RNA, and proteins. When the mRNA binds with the ribosome, a special type of RNA called tRNA, or transfer RNA, will move down the mRNA until it finds a start codon, or a specific sequence of 3 nucleotides.
In the next step, tRNA molecules will enter the ribosome and start building a polypeptide chain.
During this step of translation, known as elongation, a tRNA molecule carrying an amino acid chain will bind to the P site of a ribosome. A new tRNA molecule will bind to the next codon of the RNA and code for a new amino acid. The amino acid chain will then
Golden Corrals carved New York strip and jumbo butterfly shrimp make me feel like a real New Yorker.
You see what I'm talking? Antonio Meucci invented the telephone and he got robbed. Everybody knows that.
The amino acid chain will then transfer to the tRNA in the A site. Next, the tRNA in the P site will move to what is called the E site, which it will exit the ribosome from. Then the remaining tRNA will move to the P site and the process repeats itself.
The final step of translation is termination. This occurs when a stop codon, or a specific sequence of nucleotides, reaches the A site of the ribosome. The amino acid chain will then leave the ribosome and coil and fold until it becomes a fully fledged protein.
The process of protein synthesis is a very important part of life, and understanding it gives you a better comprehension of the world around you.